Okay, this is going to follow up a couple ideas in class that we had on Thursday the 21st from class, and it's about that protein synthesis. So just to show you again that we've got a cell, it's a eukaryotic cell, so it's got a nucleus, and we're just going to follow the path of synthesis of 4 different types of proteins. And all of them start with information in the DNA, and we'll talk about DNA a lot more in our course later in the course. The structure of DNA and RNA is introduced in Chapter 4, and in Chapter 4 is about macromolecules, and we're talking a lot about proteins and learning a lot about proteins and not so much about DNA and RNA in the course at this time. So when it comes time to study for our midterm, that information is there, but you just wouldn't study it very much because we'll study it a lot more, and we'll go back to that later. So we start out, we just say, okay, so there's DNA, and you probably have heard that the DNA is organized into genes, and there would be a gene for each of these 4 examples. So we start out with that. It gets copied into RNA. The RNA comes out of the nucleus. It's referred to as messenger RNA because it's in bringing the message of the protein code for each of these proteins. And so it's mRNA, and the mRNA is grabbed by ribosomes. And so in Chapter 6, ribosomes are introduced, and there are ribosomes in every cell. Every cell on our planet, whatever it is, has ribosomes, and they are doing an ongoing job of protein synthesis using RNA as a template. So RNA guides it because we have floating in the cell 20 common amino acids, and those are going to be grabbed using the mRNA as a code template. They're going to be grabbed in turn by the ribosomes. The ribosomes are going to synthesize proteins one at a time, and they're going to synthesize them, and the job of the ribosome is simply to form the covalent bond, the peptide bond, that creates the polymer of this protein. And each of these proteins is really similar in that they use these 20 common amino acids, but it's really the pattern, the order that they're added. And each of the 20 amino acids has a side chain, or R group, that is unique. And so the difference between the 20 amino acids is this side chain or R chain that has unique chemistry. And it's the chemistry that we've learned about. Some are hydrophobic and hydrophilic, some have a charge. And that creates different performance or different chemical behavior in this water environment. Whether it's inside or outside the cell, every cell exists in a water environment. And as a result, as this chain gets made, it's a little bit like cooked spaghetti. Remember that it can be floppy because covalent bonds can rotate around their axes. And so we get a kind of a kinky, flexible chain, and that spontaneously folds up. And we talked about spontaneous behavior in class, and spontaneous means like water flowing downhill. Water on top of the hill will spontaneously flow down. There's forces, and the forces are the chemical forces that the relevant forces for protein folding are the local chemical forces that we learned about, and they are non-covalent interactions. And that's going to cause the protein to fold up. As a first sort of thought, the proteins tend to be globular. They tend to be like a ball, a sphere, and they tend to have hydrophobic amino acids on the inside and hydrophilic on the outside. Why is that? Because it's in a water environment. And so those proteins are going to fold up spontaneously, and it's really interesting to see that they're made from the same 20 amino acids, but we get very different protein shapes and functions, structures and functions, depending on the order of amino acids. And a protein is typically a few hundred amino acids long, so we're talking about that kind of length. It will have thousands of atoms forming this molecule that's about the size of a truck driving around in Boston. And so we just follow the synthesis of this because each cell has to make what it needs for proteins. And the 4 proteins that we followed in class and in a previous video are just, and we're just going over this one more time quickly, are just that some proteins are going to head to the cell surface, like aquaporin, or they're going to head to the cell surface because they're going to be released, like insulin, which is going to be floating outside of the cell in our blood. And this is a process called exocytosis that will deliver these to the cell surface whether it's a membrane-bound protein or a secreted protein, and it goes through a process of having a free ribosome start the synthesis, and then because these proteins have a signal peptide, a signal sequence, they get delivered to the rough ER, and it's rough because ribosomes decorate the surface, and the synthesis is completed in the rough ER either by releasing the protein like insulin into the lumen, the water liquid content inside this membrane-bound organelle, or the protein will be in the membrane. And then vesicles pinch off by budding, and they will move to the cell surface, and they'll move to the cell surface through the Golgi complex, which is a place where they'll get protein modification can be made, and eventually they will be vesicles that fuse with the cell surface in the process of exocytosis. Then we also talked about that another protein that has a signal sequence is one that actually gets delivered to the lysosome, and it's the proton pump in the lysosome. And lysosomes have acidic lumen, so they're acidic like our stomach, and they are going to just digest contents of it. Oh, what happened here? And so, ooh, this is a little bit tricky. Let's try this. Well, we're going to erase some of this stuff, unfortunately. Well, it'll give us a clear space anyway. So we've got a nucleus, we've got the RNA coming out of the nucleus, bound to the ribosomes. Many are delivered to the rough ER to head to the cell surface, either to be aquaporin or insulin, and many other proteins that fit into those categories. Or a lysosomal proton pump that is going to get delivered over here, and is going to pump protons, hydrogen ions, into the lumen of the lysosome to make the lysosome acidic, low pH. And so that's a good protein to see because it also gets delivered here as a result of having a signal sequence here. So its signal sequence also gets it delivered to the rough ER. The lysosomal proton pump is first made here, not doing its job yet, so not activated yet, and we won't go into why, but inactive, but synthesized there, and then going through the Golgi, and then when it comes out of the Golgi as a vesicle, signals cause this vesicle to move in this direction and head to the lysosome. Okay, so that's that. And one of the reasons this is a good candidate is because this is another organelle, so we can see the function of this organelle. It's going to come up a couple of times, and then here we have this. We have this this pathway. And just as a side note, there are people that are born, humans born with diseases called lysosomal storage diseases. And what happens in those people is that they have a mutation in the DNA for a gene like the proton pump, and the proton pump doesn't do its job well, and so as a result this lysosome is not very acidic, and it doesn't digest or metabolize, break down these components, and so people who have lysosomal storage disease get an accumulation of this. And there are a variety of lysosomal storage diseases, and I just want to bring up one point because it's an interesting concept, and that is that some of the people have mutations in like the functional lysosomal proton pump, but others have mutations in proteins that whose function is actually in the traffic or the pathway. And that's the first time we see that, this concept of this pathway, and that there are multiple proteins in the pathway that are involved. Their function is to get things to their destination, like that. So there are a variety of proteins whose function is to work on these trafficking pathways. And there's an example, and some people are defective in this, and they simply can't get this to proper location, like that. Just a quick addition of that is there are people who have cystic fibrosis. And cystic fibrosis is a problem with a protein, a cell membrane protein, that lets ions in and out of the cell like that. And so that protein needs to get to the cell surface. It's another example of an aquaporin-like channel protein. And in some cases, people have a mutation in a gene for this protein. But in other cases, they have mutations in a gene that is essential to get this to the cell surface. And so tragically, they may make a functional protein, but it just doesn't get delivered to where it needs to be. Okay, so that's just a little addition. And but we're staying focused on our three here: aquaporin, insulin, and this lysosomal proton pump. And our fourth in class was, in a previous story, hemoglobin. And hemoglobin is a good example because hemoglobin is one of many, many proteins that's just floating free in the cytoplasm. It gets made by ribosomes, and they have the hemoglobin RNA has no signal sequence. So the ribosome just synthesizes it and lets it go in the cell. And so we're going to stop there, and we're going to pick it up in the next video.